Good morning. My name is Katrina McGuigan. Today I'm going to present to you some work that's been done by a PhD student who's finishing up in my lab, Chrissy Miller. But before I get started, thank you to Deepa for organising this wonderful symposium. I'm really disappointed that I wasn't able to get away and make it over there to join in person, but I'm looking forward to hearing the talks. Also, thank you to Howard and his team for being able to accommodate these remote presentations. They're doing amazing work organising this conference. Genetic differences amongst individuals and amongst taxa will contribute to incredible diversity in appearance, behavior, physiology, etc. As an evolutionary biologist, I feel very privileged to be able to benefit from the work of amazing researchers that have gone before me and have access to tools they developed to allow us to predict the evolution of phenotypes and to determine the contributions of adaptive versus neutral processes to patterns of phenotypic variation that we observe. In contrast to that, we have a much more limited understanding of how the evolutionary processes that generate the phenotypic diversity work to shape the underlying genetic variation of those phenotypes. And that's not because the work hasn't been done, it's just a much more complicated question to address. This is a quote that comes from the end of one of my favourite chapters in Bruce Walsh and Mike Lynch's amazing book, which having looked at all of that literature, they arrive at the conclusion that some of the empirical estimates that we have access to are a bit fuzzy. That's really where my research is focused at the moment, is trying to extend our empirical understanding of the evolutionary processes and particularly the contribution that mutation makes to phenotypic variation. And so today what I'm going to do is present some work from this experiment in zebrafish. And as I go through an outline of the design itself and the data collection, I want to kind of touch on some of the rationale for the specific design that we're working with. I'm going to start with thinking about the mutagenesis itself. And so ENU is a commonly used mutagen and it's used in forward genetic screens in biomedical model organisms such as zebrafish. In that approach, the goal is to induce as many mutations as an organism can tolerate to most efficiently, effectively be able to characterize gene functions. We've been working to try and use ENU in a different way, where we're inducing a very small number of mutations. So classical approach to looking at mutational effects is to use spontaneously arising mutations, but to look at them requires maintaining inbred lines over tens to hundreds of generations. And those kinds of experiments are not particularly viable in vertebrates like zebrafish, which have, even though they're very amenable to being managed and bred in the lab, their generation time is just long enough and their space requirements are just large enough that it's really logistically prohibitive to maintain a large number of independent populations over many, many generations. So we apply a low dose with the intent of introducing a few mutations consistent with the type of number of mutations that are segregating within mutation accumulation lines for those long-running spontaneous studies. The ENU is known to predominantly introduce point mutations and much more rarely small indels. That, broadly speaking, is a pattern that's consistent with spontaneously occurring mutations. But more data is needed to refine the shared and unique spectrum of spontaneous and induced mutations in the organism that we study. So with that corollary, I'm going to keep going. So in this experiment, then what we did was we exposed 50 male zebrafish from a particular strain WIC to the mutating agent ENU. We allowed them a couple of weeks to regenerate sperm, so we were looking at non-mosaic mutations, and bred them with a female from the same base population to generate those 50 full sibling families, which were then reared in 2 different tanks. Now, WIC was founded by a single pair derived from India. and has been maintained in captivity at relatively low effective population sizes, but it's certainly not genetically homogeneous, as molecular genetic studies have shown. So to allow us to infer the action of new mutations, we needed to have a matched set of control families. And so these are bred from males and females from the same base population, but the males were not mutagenized. Same, again, we had 50 families, and the 200 tanks in total were randomized across the recirculating water systems that we have access to. Okay, so that's the experimental populations. The trait that we're particularly interested in then is swimming speed. And we assay that using a step velocity test in flumes like this oligo flume, where water is sent around at increasing speed so that we can determine just how fast those fish can swim. Much of the literature on the effects of mutations has focused on fitness, and there's a really good reason for that. It's the effect of a mutation on fitness that will determine the persistence of that mutation in the population, what frequency it arises to, and so how it can contribute to standing genetic variation, and how it can contribute to adaptation versus extinction of populations. But nonetheless, when we're looking at biodiversity and when we're thinking about predicting evolutionary responses in populations, we're usually focused on specific phenotypes. So what effect does mutation have on those other traits rather than just on fitness? The standing genetic variation in those traits is going to be determined by the effects, ultimately by the effects of mutations on the trait itself and pleiotropic effects of it on fitness, irrespective of whether there's a direct contribution of the trait to fitness or not. So I think we need to understand a lot more about how mutation introduces quantitative genetic variation to these complex phenotypes. I'm not going to be able to do it justice in this talk, but swimming performance in fish is locomotion in fish is really fascinating. There's evidence that swimming speed can undergo very rapid evolution, and evidence that speed has evolved in natural populations in response to changes or differences in predation regime, the type of food sources that are available, and the hydrodynamic of the water flow in the environment that they encounter. There's also considerable evidence that the same types of locomotor performance or swimming speed habitat associations have evolved independently in different populations. So we have these replicated independent evolution of the same associations. And while that's considered to be strong evidence of local adaptation, it also raises those questions about whether that's reflective of a limited range of potentially adaptive outcomes, or if it's reflective of something about the way in which variation exists for swimming speed. What are the effects of mutations on swimming speed versus other aspects of fitness? Locomotive performance is something that we're particularly interested in because of the fact that it's going to be determined by morphological, physiological, behavioural traits, which can also contribute to other performance functions. But I'm not going to go into that in any more detail today. The final aspect of the experiment design that I need you to know about is the environmental context. And so we assayed these fish over 6 different temperatures, from 16 degrees to 31 degrees, where 28 degrees is the temperature that they have been maintained at in culture for the history of their domestication. It's not known what the natural thermal environment they experienced in the wild before they were captured was, but zebrafish in general are known to occupy a very wide thermal range. And they're found in shallow, no-flow water bodies, which suggests that they probably experience, some populations will experience quite strong fluctuations in temperature as well. And so in ectotherms, traits like swimming performance exhibits sensitivity to the thermal environment. So this is a classic thermal performance curve where swimming speed in this case is increasing as a consequence of temperature increasing until it reaches a certain point, after which it begins to decline, and the decline is usually doesn't make it all the way down. They kind of stop being able to function altogether, although in this situation they haven't reached that point. These thermal performance curves, again, not just for swimming speed in fish, but more broadly, are known to undergo adaptive evolution. And so they evolve in terms of changes in the temperature at which the maximum performance is achieved, and they can change in the range of temperatures over which a certain high level of performance here, fast swimming, is maintained. We know that the evolutionary divergence in those relationships then tells us that alleles don't have constant effects on the traits. So it's a non-constant effect of genotype on swimming performance. The effect is dependent on the thermal environment, but we actually know very little about the effects of mutations across environmental gradients, particularly thermal gradients. And so then in this experiment, what we did, we ended up with about 600 males. So that's from each of the 50 mutant and 50 control families, the 2 different replicate breeding tanks, and 3 sons per tank. We were focused on males because previous work suggested that males, there's larger effects of mutations in males, so more opportunity to be able to detect those effects. Each of these individual fish then was swum at each of the 6 assayed temperatures. We ended up with data from 3,479 different swimming performance trials. And before I go on to actually show you then the results of this work, I just want to pause for a moment and deal with acknowledgements. Although this work was led by Chrissy as part of her PhD research, it took a lot of work to be able to conduct all of those performance assays, as well as to rear all of those fish. And so there were contributions from volunteers in the lab, as well as research technicians and postdocs in the lab, to be able to collect that data in a timely manner. Okay, so the first thing that we needed to establish before we could start looking at some things that would be much more interesting was whether or not the mutagenesis treatment worked. We were aiming for a low number of mutations. Did we induce any increase in genetic variation? In this design, where we're looking at full siblings, then our measure of genetic variation is the among family variance. And so here is the among family variance estimates at each temperature in the control family, and this dotted line here is 0. And so for 4 of the 6 assayed temperatures in the control population from WIC, we had no evidence of genetic variance. And even though these 2 other estimates are not 0, their confidence intervals well and truly overlap 0. And so this is not inconsistent with previous work or with the long history of WIC being maintained at relatively low population sizes. In contrast to that then, when we look at the variation among the 50 mutant families, we have strong statistical support from multivariate analysis for the presence of mutational variance, and we also have support for there being a statistically significant difference in the magnitude of among family variance between the mutant and control. For this univariate presentation that I'm giving you with just the variance at each of the temperatures, in the mutants only, the variance at 24 degrees was significantly distinct from 0, which didn't survive multiple testing correction. But from the multivariate analyses, we had really strong evidence that there was among family variance in swimming performance. Okay, so then moving on to the really major question, the first of the major questions that we really wanted to try and get at with this experiment, and that is, does mutation have a directionally biased effect on swimming speed or on swimming speed at different temperatures? And so one of the challenges to being able to ask this question is having an evolutionarily inert ancestor. that's not tens to hundreds of generations removed from your mutated population, and where you can be certain that ongoing opportunity for mutation selection drift has not shifted the phenotype in that comparison population from what the true ancestral value was. That challenge is really alleviated in mutagenesis studies because of the very short period of divergence between the ancestor and the derived mutated populations, in this case just a single generation. And so this is the data, then the results from that study. And so what I'm showing you again is the temperature across the x-axis and the swimming speed along the y-axis. You can see the data follows that classical pattern where we get that increase in speed up until a certain temperature and then a decline. So I think that you can tell by eye just from looking at that plot of the data that there is a really significant difference in swimming speed depending on temperature, but there's no difference in mean speed between the mutant and control populations, and there's no difference in speed at temperature for the mutant and control. So no evidence that they have shifted, that mutation has shifted that curve shape at all. And so, we know that the effect of mutations on fitness itself predominantly seems to be biased. It's going back a while now, but the review from Halligan and Kitely back in 2009 showed that the estimated difference between the mutant and their ancestor population was typically negative for fitness effects, so fitness declined on average, and many fitness-related traits also show directional bias, like there's evidence that development time seems to be extended when in response to mutation. So the average effect of mutations has a biased effect on a lot of traits that are closely related to fitness or fitness itself. We're not seeing it at all with this swimming speed, despite the fact that we know that speed is evolves quite readily and frequently under selection due to its effect on fitness or its shared pleiotropy basis with fitness. There's been one previous study that I'm aware of that's looked at the effect of mutation across a thermal gradient, which was in Drosophila serrata, looking at spontaneously arising mutations. And there they did find evidence of a directional bias. And so mutation increased the mean locomotor activity across almost all temperatures, but not at the very hottest temperature where it significantly decreased it. And so quite a different result to what we have here for these fish. There's no temperature involved, but just looking at mean performance, other work by Huey and colleagues in Drosophila and melanic aster spontaneous mutation accumulation lines found that for larval but not adult locomotion, there was a directional bias of mutations, and it went in the opposite direction to the predominant pattern from the Latimer study. Just briefly, we've looked at before in a small number of families, just 20, at swimming speed at just 28 degrees in this population of wick, and we found they seem to find a relationship whereby in the mutated but not the control populations, speed declined with age. We looked for evidence of that kind of pattern here, but it's much more complicated to find due to repeated measures. So fish that are older also are more experienced and had training. They're encountering different temperatures because the order of temperature encounter was randomized across found individuals in the population. And so there's no evidence that anything was going on, but we have much less ability to detect it in this kind of experiment. Okay. And so the final major bit of experiment that I want to talk about is how mutation effects might depend on environment. So there's no mean effect, but that doesn't mean that that average was in reflection of mutations having consistent effects across environment. It could be mutations having effects only in some environments and not the others. So when we think about genotype by environment variation, we think about this classic kind of differences, non-parallel slopes. So if we measure a bunch of different genotypes, their phenotype in two different environments, we can look at whether or not there's a difference in how much of a change there is in phenotype between environments. or if there's this crossing over where some genotypes have got high values in one environment but not the highest in other environments. So this is the kind of information that we're really looking at, but remember we've got a more complex design than two environments. So this is what the data really looks like. This is the model corrected, smoothed representation of those thermal performance curves for all 50 mutant and 50 control families. You can see that there doesn't look like there's a lot going on. And this data, even though I've said it's model corrected, because of all of the complexity of this, really the time it took to collect this data, multiple flumes, all of the different temperatures, differences in ages and experience of the fish, different times of day, etc. The model that we're fitting is reasonably complex, and it centers the X and Y axis of this relationship in odd ways to look at the, well, not in odd ways, in sensible ways, but it's well known that how you center those axes can impact on what you estimate as the variance and covariance from random regression approaches. And what we're particularly interested in when we started this experiment as well is trying to understand whether or not the effects of mutations might result in more or less mutational variance in certain environments. And so what we did instead was look at, predominantly focused on looking at what is a character state approach, where we were estimating the mutational variances in each temperature and the covariance between temperatures, effectively treating temperature as an independent trait. So from that, we get the 6x6 mutational variance covariance matrix. And I'm not going to show you the matrix, but what I'm showing you is the parameters on this plot. And so remember, in black here is the controls. We have no evidence that there's any genetic variance in the controls. For the mutant population, consistent with what you can see here, there is all of the mutations, mutational variances and covariance, all of the mutational covariances are positive. So families that tend to swim fast at one temperature will also tend to swim fast at another temperature. Families that swim slow at one temperature will tend to swim more slowly at a different temperature as well. And consistent with the fact that we had weak evidence, very weak evidence that there was mutational variance at each of the 6 temperatures, but strong evidence that there was overall mutational effect on swimming performance, we find that 7 out of the 15 mutational covariances among temperatures were statistically distinct from 0. So we're getting a really strong signal of shared variation across the temperatures from this data. So just wrapping that up really briefly then, there's no evidence of mutation by environment variance in this experiment. We have one single dimension of among family variance that's supported, and this plot is just showing you the contribution of variation at each temperature to that single dimension that's supported. You can see that there's, they're all positive, they all go in the same direction, reflecting those positive covariances. There's a stronger contribution from 24 degrees where we did have an elevated magnitude of among family variance, and the smallest contribution from 16 where there was much less phenotypic and genetic variation in general. But nonetheless, our evidence suggests that mutations have consistent and equal effects across all temperatures. So just a general overall wrap up then briefly. I think one of the things that this experiment has shown us is when we're measuring the same thing multiple times, it really improves our power to detect mutational variance. And that's a particularly useful tool potentially for trying to get better empirical insight into these things that have really small effects on phenotype. We found that mutations had environmentally consistent effects across all temperatures, and that's consistent with what was similar to what was found previously by Latimer et al. In Drosophila serrata, where we saw that most of the mutational variance again was that kind of you're fast or you're slow, doesn't matter what the temperature is, although they did find other mutational effects in that experiment. I finally just want to note that here we're just looking at a range of non-extreme temperatures, and we're looking at acute exposure in adults. This result is not the end of the story. There is a lot more to learn about the thermal mutation by thermal environment interaction effects. And also we're only looking at the effects in F1 heterozygous offspring. They carry one copy of these mutations. We don't know whether that distribution of effects reflects the homozygous effects of the mutations or not, if there's non-additivity in these mutations. With that, I'm going to stop and thank you all for your attention, and I'll look forward to any questions that any of you might have for me. Thank you very much.